Welcome to the Beer Taster's Notebook, the show dedicated to learning about beer. I'm Jonathan Thompson, advanced cicerone, beer judge, home brewer, and beer writer. Welcome to the Beer Taster's Notebook, episode 9. We are back after the holidays. So, in keeping with our trajectory on yeast-derived aromas, we are talking about phenols. So beer always contains some sort of phenol. Of course, the levels in relation to threshold are really what matters here, especially for the aromatics. And then just to point out, there is a difference between polyphenols and phenols in this sense. The phenols we're talking about are aromatics. The polyphenols are, of course, molecules involving multiphenols, and tannins are the classic example there. And so just to define a phenol, so phenol is an aromatic organic compound. This refers to the structure. So basically, it's an unsaturated ring. Strangely, it doesn't actually mean that you can smell it, although, of course, in the case of phenols, we can, which is why we're talking about them. But it's also called carbolic acid, phenolic acid, benzenol, and hydroxybenzene. Phenol is a molecule with a phenyl group attached to a hydroxy group. It is also crystalline and can cause burns. Okay, so wait a second. We don't want that in beer, right? Well, the good news is phenols and phenol are different. Whoever came up with this should probably be shipped off to a phenol colony. Zing. Sorry. Okay, so for phenols, they're organic compounds whereby one or more hydroxyl groups, so that's an oxygen and a hydrogen, are bonded to an aromatic hydrocarbon group. Polyphenols have a bunch of phenol units and are included in phenolic acids, flavonoids, and tannic acids. So what we're concerned about are volatile phenolics. Now, a lot of the work on phenols in beers focuses on this age-old debate of health, and specifically on the antioxidant qualities of certain polyphenols, flavonoids, etc. How these various compounds contribute to stability, which of course improves storage, and how at higher levels they can add astringency, and often beyond the desired levels, and at too high a level can cause that dreaded hop burn. If you want to learn more about hop burn, there's an article on the site, beertastersnotebook.com. But for today, we're not focused on polyphenols. We're not focused on antioxidants. We are focused on volatile phenolics. Okay, so the key ones to know are 4-vinyl guaiacol, also called 4VG. That's clove, sort of spicy, nutmeg, sometimes a bit of pepper. And that is the signature clove aroma in Hefeweizen or Weissbier or Weizen, however you'd like to call it. 4-ethyl guaiacol is a sort of a smoky, woody, spicy clove and vanilla aroma. 4-ethyl phenol is that leathery, smoky, phenolic barnyard. 4-vinyl phenol is spicy and peppery. Tyrosol, hydroxytyrosol, kind of an alcohol type of aroma. Guaiacol is smoky like bacon. Chlorophenols are a Band-Aid, plastic, medicinal type of aroma, and that comes from chlorine in the water reacting with phenols. We can talk about that later. Eugenol is a clove or floral, sometimes actually a cedar aroma. Syringol is like a wood ash smoke. And then 4-methyl guaiacol is like an old greasy fire, really. And then finally, just want to mention, iodophenols are iodine related. They smell like iodine, and those come from sanitation issues. And so the precursors to these compounds are generally ferulic acid and p-cumaric acid, which come from the barley. Certain polyphenols can come from hops and then can be obviously biodegraded, but the primaries are going to be coming from the barley. And the yeast strains really do matter here, by the way. Smoking malt will add smoky phenolics, and in the very specific cases, water is a contributor. So chlorine and chloramine in water are really bad. You will end up with that the chlorophenol aroma, and it is terrible. So in some beers, phenols are not only desired, but they're actually essential. And in many others, they're a fault. So of course, it depends on the compound as well. But the chlorophenol is always a fault, of course. The 4 ones that we covered at the very beginning are the main ones. Okay, so let's dig in here. So this, by the way, is going to get way more complicated than our previous aroma subjects. So, I mean, if you want to think about esters, there are 2 components, right? An alcohol and an acid. There's a catalytic enzyme, and then the lack of a destructive enzyme, I guess, is key as well. And plus the right environmental conditions, and boom, we have an ester. So volatile phenols get way more wacky than that. But let's kind of try and go through the process. So step 1, you need a precursor, like a phenolic acid, which live in the cell walls of cereal grains and are extracted in the mash. especially if a specified rest is used. So ferulic acids and p-coumaric acids are the most common, and their rest is around 45 degrees Celsius, by the way. So then the next step involves the yeast, and very specific strains are key. So enzymes called PAD1 and FDC1, which are phenylacrylic decarboxylase and ferulic acid decarboxylase, remove a carboxyl group forming the vinyl derivative, which is then quite volatile. Clear? Okay. Not really, right? So certain microbes, by the way, perhaps because they're jealous of a lack of invitation, can further reduce these into ethyl derivatives, and then that totally changes the flavor. So let's get a little deeper here into the pathways. So ferulic acid, p-coumaric acids, caffeic acid, synaptic acid, those are 4 hydroxycinnamic acids that are found in barley, and they're bound in the arabinose islands in the cell wall. They are ester linked to polysaccharides and need to be liberated before they can actually do anything. So what this means is the acid is bonded to the hydroxyl group on the sugar unit. And during the mash, at the rest temperature of between 43 and 45 degrees Celsius, with a pH of about 5.6 to 5.8, these bound acids are liberated by a ferrolyl esterase. And so now we have these precursors, and they're fancy and free in the wort and swimming around. And the yeast strain here is now the key. So only strains that are POF or PAD positive, which are phenolic off flavor or phenylacrylic acid decarboxylase positive, will be coded for the enzymes that catalyze this transformation. So there's 2 enzymes involved here, both codependent. So there's PAD1 and FDC1. So PAD1 produces the PRFMN cofactor, which is then used by FDC1, which decarboxylates the acid. So this is a non-oxidative decarboxylation, by the way, so no ATP is produced, but CO2, of course, is released. Now, a lot of yeasts have those genes turned off, and in fact, most do. So there's a reason why it's called phenolic off flavor, right? And so the yeast, by the way, produce mostly vinyl phenols, and these can then be further reduced to ethyl phenols. The difference is between these 2 compounds is the functional groups. A vinyl has a carbon-to-carbon single bond, whereas the ethyl group will have a saturated single bond chain. The issue with some of this stuff is the colloquial names for certain things can conjure up peculiar imagery, by the way. So, I mean, when you think about vinyl, you're probably not thinking about beer, right? Or especially beer aromas. I mean, you're thinking about maybe, a raincoat or a record. And so what we actually call vinyl just in the wild there is, polyvinyl chloride. Acrylic also suffers the same affliction. So just to put those sort of out of your mind, or maybe I just put them in your mind. I don't know. Anyways, back to the plot. So the reduction can occur with Saccharomyces strains for sure, but for the most part, it's carried out by the Brettanomyces strains. So the reaction is carried out by the vinyl phenol reductase, and then that requires NADH, and often occurs in really low oxygen conditions. So in fact, anaerobic conditions boost phenol production in general as is higher temperature. And while we're on the subject of conditions, overpitching will suppress production, while underpitching can cause yeast stress and then push phenolics up. Low fan also can boost phenolics as well because it is an additional stressor. Okay, so back to the reduction. So the vinyl phenol is a ring with a vinyl side chain. Double bonded carbon, as we mentioned. And so the vinyl groups have a few features. They are electron rich, and so they're very ripe for hydrogenation. So the basic reaction is, and we're going to use 4VG just because it's the most common. So the 4VG plus NADH turns into 4-ethyl guaiacol, or 4EG rather. Yes, not rather, but the same, plus NAD plus, and then so it's a 2-electron reduction. So we don't really need to go much deeper than that because it's not super important for this purpose. And it's almost, I don't want to say it's impossible, but it's going to be really difficult to explain this without some visualization. So this is carried out by VPR enzymes, vinyl phenol reductase enzymes, which are oxidoreductase enzymes. I guess I just said enzymes a million times there. That's fine. It transfers hydrides from the NADH protonating the vinyl group and then saturating the double bond. So the NADH binds the enzyme. The hydride is transferred to the beta carbon. The proton is added to the alpha carbon, and the vinyl group becomes an ethyl group. Okay, I guess I said I wasn't going to do it, but then I did. But anyway, there you have it. Maybe I'll cut that out later. So just some interesting notes about phenol and ester interaction. So both pathways compete for NADH and acetyl-CoA. So the higher the esters, the lower the phenolics and vice versa. This explains, of course, that big array of balances in Hefeweizens. In fact, I put out a little piece. It's pretty much Ontario specific, to be honest, because it's what's available at our retailers. But just on the different profiles, aroma profiles of the various different commercially available Hefeweizens. But I mean, it is interesting. It's just though the small difference can actually make a massive difference in sensory. Okay, so before we get biochem overload here, let's talk about why yeast are making these phenolics in the first place. So, contrary to how we might want to feel, yeast have no interest in providing us with delicious flavors. They don't care. Phenolics are antimicrobial, so it's kind of a defense mechanism. And so there are a few other things come into play here as well. NADH is important too. So it's a product of glycolysis, amino acid catabolization, and anaerobic fermentation. And so the cells must regenerate NAD+, or the metabolic process fails and everything falls apart. So I will do a little metabolism sort of primer separately down the road, kind of like a reference episode. But basically, redox is central to biochemistry. And so the constant cycling of NADH to NAD plus to NADH ad infinitum is key to pretty much everything, really. So aldehydes are reduced to alcohols using NADH and then spit out NAD plus. The same thing with the reduction of vinyl phenols to ethyl phenols. And so yeast, at least standard Saccharomyces, really favor ethanol production as a way of regenerating NAD plus when they're in specific conditions. And they don't have a particularly strong VPR genes. Brettanomyces, on the other hand, has highly expressed VPR genes. It is very comfortable in low sugar and low oxygen environments, and it is often subject to NADH pressure, triggering more reliance on alternative electron or redox sinks. So a redox sink, by the way, is a molecule or group that accepts electrons. Generally, it's oxygen in the electron transport chains, but if oxygen is not available, obviously we need an alternative. So vinyl phenols become this preferred sink, and so reduction to ethyl phenol occurs. Brett eats very, very slowly, which is why the longer a Brett inoculated beer sits, the funkier it gets, right? And so the major feature for us, anyways, of this reaction is this dramatic shift in flavor. That reduction is just one carbon bond, but it makes a massive difference. On to some of the other microbes. Lactobacillus contributes very little in the way of phenolics, but Pediococcus can bring something very interesting. So it does not code for the PAD-FDC pathways, and then therefore doesn't produce the compounds via phenolic acid decarboxylation. Instead, it produces the phenols via phenolic acid metabolism or reduction of the hydroxycinnamic acids, ferulic or p-coumaric, of course, or via amino acid catabolism, which results in p-cresol, which is really bad. That's actually a huge fault. It's terrible. So a third pathway is stress-induced. And so when pH gets low, ethanol gets high, and oxygen drops, this sets up for redox pressure and then results in ethyl phenols, albeit fairly low. Where everything gets quite crazy is if pedio teams up with Brett. And so the Pediococcus liberates these acids and precursors into intermediates while lowering the pH. Brettanomyces then comes along and converts those intermediates into 4-EP, as well as reducing the vinyl phenols to ethyl phenols, and then the barn doors are blown wide open. I think about lambic beers and Geuze and stuff. It's the teamwork that makes that funky dream work between Pedio and Brett. And so on their own, Pediococcus phenols are generally not desired. So reduced phenols can smell smoky or plastic-like, and p-cresol smells like antiseptic, as do the hydroxyphenyl propionic acids. So it's just a lot of that stuff is not what you want. Okay, so just to wrap up here, so the production of detectable levels of phenolics is dependent, yes, on inputs and conditions, but is wholly dependent on the yeast genetics as well, and whatever other microbes might be in there. So yeast described as POF positive have the genes to code the required enzymes turned on. Longer yeasts, clean American ale yeasts, have this gene mutated out. In fact, that's very common in a lot of yeasts. Saison, Weizen, Belgian ale yeasts all tend to be POF positive. Kveik strains, as well as some others, and I'm using quotations here, farmhouse strains, will have that gene intact. And so obviously condition control is key to whatever flavors you're looking for. Okay, so let's just sort of do a recap of what phenols are what. So the major, of course, is 4-vinyl guaiacol, 4VG, yeast derived, common in Weizen and saisons, and to a degree, Belgian ales. Smells kind of like clove and or nutmeg. 4-vinyl phenol, 4VP, that's the one described as spicy, but it can seem medicinal at higher levels. And this is the precursor, of course, to 4EP. 4-ethyl guaiacol it's kind of like smoky bacon. It's a little bit clovey still. It is the reduction of 4VG by Brett. 4-ethyl phenol, 4EP, so that's the classic Brett character. Leather, barnyard, horse blanket. By the way, I've smelled a lot of barnyards in my time. And recently I smelled some horse blankets, on purpose. I asked my sister if I could smell some of her horse's blankets, and she kind of just stared at me, but eventually said okay, and was really curious as to why. Anyways, I can tell you this. I understand the parallels between these descriptors, but we can do a lot better. If you get the chance, open up a lambic beer, preferably not one that's super fruited or any kind of Brett beer, right? Go find a horse blanket, and I do realize this is kind of a crazy situation, but find a horse blanket and smell them side by side, and then report back and tell me that that's exactly what they smell like. There's some similarities, I understand, but we need to do better with these descriptors. Anyway, there's my rant for today. So where are we here? So guaiacol comes from smoked malt directly, as does 4-methyl guaiacol, which is sort of signature in the Rauch beers. P. cresol, as discussed, is a hospital smell. The O and M cresols are more like tar and smoky, respectively, but they're also not something you want. Chlorophenol, which is the result of poor water control. And it's a subject actually we're going to tackle down the road, but it smells like plastic and also Band-Aids. And that's an easy one. So seriously, pick up a Band-Aid, like a new fresh Band-Aid, and give it a smell, and then you smell a beer with chlorophenol. And that is a very accurate descriptor. There are definitely more out there. So we could just go on and on and on, but we won't. And so those are the ones we really want to know about. Actually, no, that's not true. So ethyl catechol and ethyl syringol are Brett derived, and they are sort of earthy, woody, and smoky. So you should probably be familiar with those as well. Okay, now I'm done. Okay, so that's phenols down. Next week, we are going to tackle thiols, which will bridge the gap into hop aromas. They're big, tropical, and they're exciting. If you find this podcast useful, helpful, or just exciting, please remember to rate, like, or follow. Thank you. Until next time, stay thirsty and stay curious.